For the next presentation is study on the role of ferroptosis in cardiac injury induced by obstructive jaundice by Zhezhe Fan from College of Basic Medical Science, Naval Medical University. Please start when you're ready.
Good morning, ladies and gentlemen. I'm Fan Zhezhe from Naval Medical University. Today, my topic is study on the role of ferroptosis in cardiac injury induced by obstructive jaundice. I will elaborate on the following four sections. The first one is background. Obstructive jaundice, OG, is caused by bile duct obstruction, leading to impaired bile excretion, bioaccumulation, and reflux into blood. Beyond the clinical symptoms like jaundice, OG causes OG causes severe damage to multi-organs, for example, liver, kidney, heart, and intestines. Biases are almost implicated in these mechanisms, and they are particularly damaging to the heart, including bradycardia, arrhythmia, and even heart failure. So, understanding the link between OG and cardiac injury is significant. As I said earlier, biases play a key role, and our article reveals that it's particularly damaging to the heart through 8 primary pathways. Notably, oxidative stress is significant due to its link to ferroptosis. What is ferroptosis? It is an iron-dependent programmed cell death driven by increased reactive oxygen species and iron accumulation, which is tied to oxidative stress. And it's characterized by lipid peroxidation and the loss of antioxidant capacity. So we hypothesize that OG-induced cardiac injury is mediated by ferroptosis. Next is methods. Firstly, we divided the mice into 4 groups by building BDR models and administering ferroptosis inhibitor. For study 1, we observed ferroptosis and cardiac injury occurrence in each group after 7 days. This step was primarily verify whether the OG-induced cardiac injury is associated with ferroptosis. After confirming that, we conducted the second step of our project. In this step, we investigate which of the 3 major signaling pathways was involved. And based on our preliminary results, we focus on GPX4 and validate this hypothesis by building GPX4 overexpressing mice. And after that, we got the following results. Firstly, we found that OG mice showed significant weight loss and liver injury. Some exhibited abdominal jaundice and ascites systems. This demonstrated that the remarkable success of BDR model establishment. And secondly, OG influenced cardiac functions with OG influences cardiac functions, including slow heart rate and ventricular remodeling, and it can lead to changes in ejection fraction, fraction shortening, and cardiac output. For study 1, partially restored them. Moreover, cardiac tissue in OG mice showed increased MDA and iron levels, but GSH and GPX4 expression were decreased, and immunofluorescence revealed that increased 8-OHDG expression in OG mice. For study 1 can reverse these changes, and this indicates that ferroptosis is deeply involved in OG-induced cardiac injury, and the GPS4 play a pivotal role. So we conducted the second step of our project, and we found that after overexpressing GPX4, the ferroptosis-associated markers were decreased, like ACSL4, and in addition, the staining section showed inflammation levels and the fibrosis degree of myocardium were decreased, and myocardial injury was improved too. Lastly, we found that GPS4 overexpression improved cardiac functions with recovery in ejection fraction, fracture shortening, cardiac output, and heart rate. Based on the bioanalysis, we think we provide a deeper understanding of how ferroptosis and OG-induced cardiac injury are connected. After OG, increase bile acids act on BA receptor, downregulating GPX4 expression, triggering ferroptosis, and finally causing cardiac injury. We think aiding a bile acid receptor antagonist group is promising based on our mechanism. Our project was supported by our university fund, has been successfully concluded and received several awards in competitions. And our review is under review. Thanks for your question. Thanks for your listening. All the questions are welcomed.
So in your GPX4 overexpression model, so it is a global overexpression, or it just act specifically at the heart?
Thanks for your question, Professor. Actually, in our experiment, the mice overexpress GPS4 systemically, but so the systemic improvement is permitting. But in our experiment, we found that after the use of Ferroptosis 1 and GPX4 overexpression, the cardiac function and cardiac injury were improved both. We think so the cardiac effect is real, and we think maybe the cardiac improvement is based on other organs like liver, because more studies has shown that Ferroptosis 1 or ferroptosis inhibitor can improve liver injury. But in our experiment, it may improve to some extent because we think within just 7 days, it is very difficult for heart and liver to build a strong crosstalk. More studies have shown that liver and heart may build a strong crosstalk, but in our experiment, 7 days is a very short time. So
I do agree that you have shown improvement in functions at the heart tissue, but do you have an idea if you can test whether the improvement of cardiac function is derived from ferroptosis inside the myocardial cells, or it may be a secondary effect from other organ tissues? Okay, can you discuss an idea how to investigate that?
Yeah, that's a very promising. Because in the future, we think we can aid cardiac-specific GPS4 overexpression mice or liver-specific GPS4 overexpression mice, or ferroptosis inhibitor, a liver-specific ferroptosis inhibitor mice. Thank you.
According to your presentation, it indicates that obstructive jaundice is related to GPX4, right?
Yeah.
And just only this is only one factor that induces the OJ in the human, because usually OJ could be happen by many factors.
Thanks for question, Professor. In our experiment, we observed investigate the function of GPS4 in the process of OG-induced cardiac injury, but we think that there are maybe other mechanisms, for example, inflammation and endotoxemia, because gut barrier impaired gut barrier can allow endotoxin into the circulation, which can directly affect the heart and trigger inflammation. And the second mechanism may be mitochondria dysfunction, because in our experiment we found that oxidative stress happened, and chronic oxidative stress can impair mitochondria and reduce ATP production, which is crucial for cardiomyocytes. And the third potential mechanism may be endothelial and microcirculatory dysfunction, because bile acids and cytokines can impair the endothelial cells and disturb the coronary microcirculation. Thank you.